Hey, Hammy here, back with the last video, finally, right, of section 2.1, where we will talk about the last two biomolecules, proteins and nucleic acids. First of all, proteins. Proteins are important in our bodies for a variety of reasons. We'll look at it in a little bit. If we remember back to the other video, remember there are small subunits called monomers that we add together to get big molecules called polymers. Okay, big chains of the smaller molecules. I kind of compared them to Lego bricks. The monomers in proteins are these guys called amino acids. Okay, where does the term amino acid come? If we look at the structure down here, I often joke that the base structure is North Carolina Cougars. Now I know there's no such team, but that's how I always remember it in my head, the North Carolina Cougars. And the North stands for nitrogen with two hydrogens off of it. Okay, we can ignore this one for now. Okay, we call that an amino group. The other side has a double bonded oxygen, and then another oxygen here, often represented with an OH group. This one just has lost its hydrogen here. Okay, that's called a carboxyl group or carboxylic acid. So you have amino group, carboxylic acid group, amino acid. See where that comes from? Okay, so these little amino acids here are what are chained together to make the bigger proteins. This R group, notice down here R, just like in math, when you're doing long division you put R or remainder. This is the remainder of the molecule. There are 20 different amino acids found in living things, and we can make thousands and thousands of proteins from those 20 amino acids. Just like you have 26 letters, how many words can we make in the English alphabet? Hundreds of thousands, right? So with 20 of these amino acids, and some of them simply just have a hydrogen here, others might have an OH, and some have these big ring structures that hang off the bottom of the amino acid. Okay, those 20 amino acids are used to build every single protein that living things need. Okay, and sometimes it can just be a couple amino acids. Sometimes, like the protein titin, T-I-T-N, they estimate is about 27,000 to 33,000 amino acids long. Okay, huge. It's found in our muscles, and that's one of the largest proteins that we have found in the human body. Okay. How are these things linked together? Okay, remember, we take out a water. Okay, so between the amino acid here, amino acid number 1, amino acid number 2, we take out a water, so HOH or H2O, we take out a water, and then it's going to use that bond right there. This is what we call a peptide bond. So when you combine two amino acids, now you have this down here would be a dipeptide. You start chaining a whole bunch together, 3, 4, 5, 6, 7, then you get what we call a polypeptide. Okay? And like I say, they can get very, very, very large, like the protein titin that's like thousands of amino acids long. What happens is these amino acids, okay, the primary structure, okay, you have translation happening. That is at the ribosome in the cell, where the instructions from the DNA are brought out from the nucleus, and the RNA starts putting these little round things here are amino acids, and it starts chaining them together, okay, with peptide bonds in between there. Okay, this long chain of amino acids is called the primary structure. Right after it's made, it's just a string, almost like a string of pearls. Okay, that's just the primary structure. What begins to happen is those R groups that hang off the bottom, those R groups begin to interact. Okay, and you have the similar picture in your notes. For secondary, highlight hydrogen bonding. Okay, hydrogen bonding with those R groups starts to cause interactions between the little folds and bends here. Okay, And when it does that, it can do two things. It can make a pleated sheet where it kind of zigzags, and you get little interactions there between the polypeptide amino acids. Or you can get an alpha helix where you get kind of this helical structure, okay, kind of a spiral shape. Okay, what those then begin to interact with each other, sometimes through what we call disulfide bonds, and the polypeptide, these helices and pleated sheets begin to interact with each other, and it kind of crumples up, like taking that pearl of beads, that chain of pearls, and crumpling them all up into kind of a bunch. Okay, this is called the tertiary structure. Okay, now it's got kind of a three-dimensional shape. Okay, that shape, I often tell the bio-AP kids, F cubed. Okay, where form fits function. Okay, the shape of that protein is very important for the job that it plays in the body. Okay, and its shape is dependent on these little interactions of the R groups that are hanging off the bottom of those amino acids that causes it to coil and bunch up. And then sometimes quaternary, you get several polypeptides. Okay, for this example, actually has two polypeptides thrown together. Hemoglobin, that we'll talk about here in a little bit. Hemoglobin is actually four polypeptides put together. Okay, this would be the quaternary protein structure, kind of its final structure. Several polypeptides often put together for that final protein that does a job in our body. Speaking of functions, there's four main functions of proteins that I want you to know. Number one, and most importantly, are structural. For example, keratin that you find in the skin. Okay, keratin, hair, nails, it's kind of this tough protein that helps waterproof our skin. Okay, and it helps keep out germs and bacteria and stuff like that. Function number two, regulatory. Okay, it regulates. An important one are enzymes. Okay, enzymes will control the rate of chemical reactions. You also get hormones. Over here's a picture of an enzyme. So again, you see here these alpha helices and pleated sheets, kind of these zigzags. Enzymes are needed to break down things. Enzymes are needed to put things together, build things in our body. And also hormones that regulate our body, how our organs talk to each other without using the nervous system. Number three, they transport materials. This is a big one down here. I said we'd mention hemoglobin again. Hemoglobin is a protein found in our red blood cells. They look like little red donuts almost. And there's four polypeptides. Oh, forgot three. 1, 2, 3, 4 polypeptides with little heme or iron groups. Those iron groups, okay, help carry oxygen. It's kind of small to see. They carry O2. They carry an oxygen molecule. So when you breathe in, the oxygen is picked up by these red blood cells, okay, and then carried out to your muscles, to your brain, to all your cells where oxygen is needed for cellular respiration to provide them with energy. Also transport not just things through your body, but things in and out of the cell. When we learn about cell membranes in the next chapter, we'll look at proteins that are stuck in the cell membrane, little protein channels that let stuff go in and out of our cells. And fourth and finally, we use proteins for protection. We have these little Y-shaped antibodies, okay, in our white blood cells as part of our immune system. Okay, antibodies will find and attach to antigens, which are on the surface of all other cells. So when bacteria or something like that get in us, our antibodies will recognize them and will go kind of attach to them, destroy those cells, and help defend our body against disease. Finally, we have the fourth one, okay, are nucleic acids. Nucleic acids include DNA RNA, deoxyribonucleic acid. Okay, the subunits or monomers. Monomers of nucleic acids are what we call a nucleotide. There are three things in a nucleotide. Okay, there's a phosphate group, we usually draw as a circle. There is a sugar, a 5-carbon sugar. In DNA, it's deoxyribose, hence the name. We often just draw as a pentagon, and we connect them. And then there is a base, or sometimes we'll say a nitrogenous base, because there is nitrogen in there. And these often have various shapes here that are connected here. There are 4 nitrogenous bases that you need to be familiar with. In DNA, contains some single-ringed bases, cytosine, often abbreviated with a C, and thymine, okay, abbreviated with a T. And then there are purines called adenine, which is often abbreviated A, and guanine, G. Now there's base pairing rules in this DNA molecule. A pyrimidine will always bond to a purine. In RNA, over here, you'll see it has a U, uracil, instead of thymine over here. And on the next slide here, we'll take a look at kind of how these bases interact with each other in DNA. Okay, here you see the structure of DNA. You can see that, here is the pentagon, so that is the sugar, the 5-carbon sugar. Up here is the phosphate group, okay? So it goes phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. On the other side, okay, it goes phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. So phosphate sugars make up the backbone, or the kind of like the sides of the ladder. And then the rungs in the middle are your bases. Okay? And there's these base pairing rules. Okay, and we'll learn this in the DNA chapter as well. But cytosine, okay, a pyrimidine will always bond with guanine, a purine. So C and G always bond together, and you'll see that thymine will always bond with adenine. And they can be on either side. So here adenine is on this side, thymine is on this side. Okay, and they're like the rungs. And then right down the middle, where you see these these dashed lines. Okay, these dashed lines are hydrogen bonds. Just like in proteins, when it starts to twist and coil and you get weak hydrogen bonds holding them together. In DNA, the two sides, okay, the rungs with the bases sticking in the middle, between those two bases, okay, down through here are hydrogen bonds that hold the two sides of the DNA molecule together. Okay, and then the whole DNA molecule is twisted into what we call a double helix, and that's how we often recognize DNA as being DNA, because it's twisted into a double helix. RNA would just have ribose sugar, so right here would have a ribose instead of deoxyribose, and it would, instead of T, it would have U, a uracil. Okay, so it would also just be single-stranded. RNA, there's not two sides of it. RNA is just a single strand. So that's some 3 main differences between DNA and RNA. Why are these bases important? The A, T, G, and C is basically our genetic code. The order of those bases in our DNA codes for the order of the amino acids that is used, like I said, out at the ribosome to build our proteins. That's how our genetic code translates into who we are. Okay? These nucleic acids are the basis for passing information from one generation to the next. There is one example of nucleic acid that is not used, like DNA or RNA, for genetic purposes or genetic code. Okay, it is ATP, which stands for adenosine triphosphate. And when we talk about cellular respiration and photosynthesis, we'll look closer at this. But we often call it the energy currency of the cell. A lot of books liken it to a battery. So like on our Chromebook or our cell phone batteries, we say, we charge it overnight. In the morning, it's all charged up. Okay, that charge that our body uses is right between the second and the third phosphate. And when we break off that phosphate here, it releases a little bit of energy. And this is just the right amount of energy to maybe pump things in and out of our cell, for muscle contraction, nerve impulses. ATP is just the right amount of energy for those processes. And so we call it kind of the energy currency of the cell. Okay, so this kind of wraps up our 4 biomolecules: carbohydrates, lipids, proteins, nucleic acids. Okay, please go back and rewatch those and read your textbook. And feel free to ask me if you have any other questions that you need me to explain on this.